till now we have done everything that we need to implement our project, that is the data set which we require to move further. So in the next step is to split the data that we have till the point here. So we need to split the data into two sets, that is training set and testing set. So in this project we are going to split the data in a ratio 2:1. So training data set will get 2 parts and testing data set will get 1 part of the whole data set for implementing of this project. So I am going to write 0.67 because it is two-thirds of the whole data set, and n rows indicate the number of rows. So in the total number of rows present in the breast cancer data set, I am going to floor indicate that we are converting or rounding it. So and rounding it and saving those in sample split. So let me run this. So you can see what are those in the sample split. So sample split is 457. So there are 683 approximately that are present in our whole data set. So now we will get sample split, that is 2/3 of the data set is 457. So let me seed in the code it, because when the multiple users run on different systems, this will ensure that the data is split on the same manner or on a similar manner. So after doing this, let me take the sequence length. So we'll get in sequence length indicate this will generate a sequence. So how much sequence is generated? So the variable or the value that is passed inside this brackets. So let me run this and you can see, so it has generated a sequence from 1 through 683. So 683 is the total number of rows present in our data set. So for training we need a sample split size, so 457. So when we give sample of k comma size equals to sample split, k indicate the total number of entries in the data set and size indicate the size of the number of samples that we need to sample. So when we pass these two, it will randomly sample the whole data set and store 457 values into training. So training will have, let me show you, will have indices that are sampled at random and a total of 457. So after doing that, let me take the training data into cancer train by writing breast cancer of, this is the way we index into the training data set, and I'll write all rows. So training, whatever the indices that are present in the training will get into cancer train, and when we write comma and write nothing, so all the columns are taken for each row. So let me run this. So after that, let me show you the data set. So this is the data set, you can see here. So you can go ahead and see, and now the similar process is done for taking or preparing the test data set, that is taking test data into cancer test. But in this case we will write minus in front of training to indicate that whatever the indices that are not a part of training indices, so all those which are left out are taken into cancer test. So let me run this, and I am going to remove the target variable or the class column from the cancer test and store it in cancer test data, because when we are testing a model, we only pass the features and predict on the features and compare it with the original test output that is present with us. So I am removing the 10th column from the whole dataset of cancer test and storing it in cancer test data. So let me do that. So after doing that, let me load the data. So you can see that, one second. So we have only till mitosis and class column is removed. So let me take the class column next. Only the class column into cancer test out. So cancer test out indicate the target column. So it will have the target or the actual output for the test data. So let me stop the video here. We'll meet in the next video.